Good morning, good afternoon, and good evening. Welcome to the U.S.-Japan Brain Research Cooperative Program Scientific Webinar. I'm Stacy Chambers, Scientific Project Manager in the Office of Global Health and Health Disparities at the National Institute of Neurological Disorders and Stroke. Over the next hour, this webinar will showcase some of the impactful scientific collaborations that have been supported by the BRCP, along with truly innovative brain research that is currently being supported at the National Institutes of Health in the United States and the National Institute for Physiological Sciences in Japan. As a reminder, you may submit your questions for our presenters in the Q&A chat, and they will be answered during the open discussion session after the presentations. It is now my pleasure to introduce Dr. Richard Benson, Director of the Office of Global Health and Health Disparities at NINDS, and the U.S. Chair of the U.S.-Japan BRCP. Dr. Benson.
Thank you, Stacy, for the introduction, and also thank you, Naomi. Again, good morning and good evening, everyone, and welcome to this U.S.-Japan Brain Research Cooperative Research Program scientific webinar. Just to give you a little background on the program, the U.S.-Japan Brain Research Cooperative Program promotes scientist exchange, training, and research collaborations between neuroscientists in the U.S. and Japan. The U.S. component of the program awards administrative supplements to currently active research grants that are supported by one of the participating NIHICs, and we have them listed at the top of the slide. We have NIDS, NCCIH, NIBIB, NIDA, NIDCD, NIEHS, and NIMH, and I thank all of my NIH colleagues for participating. The BRCP is a bilateral partnership with the National Institute of Physiological Sciences in Japan as well, and you can go to our website that Naomi Booker and Stacy Chambers put together and did an excellent job. The results from this collaboration and this workshop that has been going on for over 20 years, I think over 25 years now, is we have training areas and training and new techniques and technologies for people in the US and Japan. Some of the tools include molecular tools, microscopy, imaging, and electrophysiology. More than 60 US trainees have been supported under this award through collaborations and workshops. We also foster new collaborations and future research as well, and the submission of joint applications to the NIH as well, and many presentations of abstracts at scientific meetings and publications in journals as well. And so the purpose of this meeting today is to highlight some of the similar work to spark new research that's being done in the US and Japan. And again, I'd like to thank all of my colleagues here at the NINDS and the NIH as well, particularly Stacy Chambers for doing an excellent job of spearheading and leading this program over the years, Naomi Booker, and my leadership as well. I see we have several leaders. Dr. Andrea Meredith is on the line, and I'm not sure if Dr. Amy Adams is on as well. Dr. Korschetz said that he would try to make it, but he may or may not be here. So I'm going to end there, and I'm going to hand it over to my colleague in Japan.
Thanks so much, Richard. Now delighted to introduce Dr. Masaki Isoda, professor at the National Institute for Physiological Sciences and the Japan chair of the U.S.-Japan BRCP. Dr. Isoda.
Hi, good morning to Japanese colleagues and good evening to U.S. colleagues. I'm Masaki Isoda from the NIPS in Japan, and I'm the chair of the BRCP Steering Committee on the Japan side. And it's my great pleasure to hold this scientific webinar today. And as Dr. Benson mentioned, the BRCP is a long-standing initiative supported by the government of Japan and the United States. and it has continued steadily for almost 25 years. And during this period, the Japan component has supported research spanning all levels of brain sciences, from molecular and cellular to systems levels, with the goal of understanding brain functions in physiology and disease. Indeed, by supporting 230 research projects to date, we have significantly contributed to the advancement of collaborative research between neuroscientists from Japan and the United States. And today, we have invited the former BRCP recipients who are, of course, the front runners in the field of brain science. But we also have invited key neuroscientists, representative of the major brain science research projects in 2 countries, that is Brain Mind 2.0 in Japan and Brain Initiative in the US. I really appreciate their participation to this webinar. Okay, I really hope that this opportunity will help us further promote and broaden research collaboration between the 2 countries. Thank you very much.
Thank you, Dr. Soda. It is now my privilege to introduce Dr. John Nye, Senior Investigator and Director of the NIH Brain Research through Advancing Innovative Neurotechnologies Initiative, also known as the Brain Initiative. Dr. Nye, over to you.
You're on mute, Dr. Nye.
Sorry, of course, thank you, Richard. So I want to thank my colleague, Dr. Benson, for reminding me of that, but also Isoda Sensei for your organization and leadership in this program. I'm very excited to be here. So I just want to spend a few minutes telling you a little bit about the NIH Brain Initiative and how we might use programs like this to foster greater international collaborations. Let's see. So the NIH Brain Research through Advancing Innovative Neurotechnologies, or Brain Initiative, is a highly collaborative and interdisciplinary trans-NIH enterprise that's propelled by the participation of 10 of the 27 NIH institutes and centers. Our mission is to revolutionize our understanding of the human brain by accelerating the development and application of innovative technologies. This initiative was announced in 2013 with the first awards made in 2014 and represents a partnership between 5 U.S. federal agencies as well as private foundations and philanthropies. The NIH efforts have been guided by 2 strategic plans, the Brain 2025 report that was issued at the very beginning of the Brain Initiative in 2013, as well as the Brain 2.0 reports that came out in 2019, and these have been our guiding documents. Now, Brain has been in operation for just over a decade. A high-level overview of our first decade is provided in this perspective piece published in Neuron in the fall of 2024, detailing in very broad strokes the various accomplishments, tools, and resources that Brain has supported over the first 10 years. Too much to go into today. So today I want to instead highlight some of our large resource-generating projects that provide opportunities for collaboration and synergies between neuroscientists, both in the US as well as in Japan. So several years ago, we launched some large projects that are underpinning a tool-driven revolution in the neurosciences. We view the human brain as the most complex, certainly the most complex organ in the body, but also arguably the most powerful computer known to humankind. It contains about 86 billion neurons, making trillions, if not hundreds of trillions of connections with each other. In order to understand how this amazing organ computes and gives us all the properties that make us human, we really do need to know more about the components, the cellular components, how they connect with each other, the wiring diagram, and to be able to use this information and build tools to test hypotheses about neural circuit function, and very importantly, to devise new interventions for when these things go wrong in disease. So we've stood up these 4 projects. I'll just go through them very briefly, one by one. The first on the left is the parts list, basically a cell atlas of complete mammalian of whole mammalian brains. And right now we're in the third phase of the Brain Initiative Cell Census Project, now known as the Brain Initiative Cell Atlas Network. This gives us the part list, the cellular constituents of this amazing biological computer. The next is the Brain Connectivity Across Scales, or Brain Connects network, which will give us the wiring diagram. How do the cells in the brain connect with each other, ultimately giving us the ground truth for how these neural circuits underpin everything about behavior and sensation and so forth. The third project is what we call the Armamentarium for Precision Brain Cell Access, a large toolkit for accessing cell types in the brain with cell type and circuit precision, right now using viral tools, but potentially using other molecular tools that allow researchers to target cell types and circuits with exquisite precision to ask questions about how cells and circuits underpin behavior, ultimately to form the foundation for precision gene therapies for human brain circuit disorders. The final project is the Brain Behavior Quantification and Synchronization Program. This is the youngest of the 4 projects. And here the goal is to develop better tools for measuring and quantifying animal and human behavior, and then to synchronize that information. This is where the synchronization part comes in, to synchronize that information with high resolution, high quality neurophysiological data in order to start generating cause-effect relationships between neural activity and behavior, ultimately with the goal of developing closed-loop therapies for human behavioral disorders. So these are the 4 large projects in broad strokes. We have many, many other projects, but this really is in support of driving the field forward by providing large-scale tools that will enable neuroscience research around the globe. So we've been creating neuroscience resources at scale, and I'll just summarize those very briefly here. The first comprises the parts list. So we've developed a complete cell atlas of the mouse brain. We've identified over 5,000 cell types from an analysis of over 32 million cells, and this has involved extensive replication as well as cross-validation. The teams have also developed and published draft non-human primate as well as human brain atlases, which sets the stage for a comprehensive understanding of the human brain at the cellular level. The Brain Initiative Cell Atlas Network recently published in Nature a package of papers describing a developmental atlas, the cataloging and understanding of the brain cell types as they appear during development, which provides a framework for understanding the specification and development of brain cell types and the origins of neurodevelopmental disorders. So we're very excited about these projects. In terms of the connectivity project, there's been a lot of progress here. This shows you the results of the Flywire project. This is the first complete connectivity map at synapse resolution of a whole complex adult animal, that of an adult female Drosophila or fruit fly brain, which is about the size of a poppy seed. But in this very dense network are 139,000 neurons, comprising 4,500 unique cell types based on morphology and connectivity, making over 50 million synapses with 115 meters of axons. And the beauty of this project is that it's been open source. It has led to the ability of researchers in Drosophila to use the ground truth information about the connectivity map to inform and constrain models for neural circuit function. And it's also set the stage in terms of technology for standing up larger connectivity maps for example, in mammals. So here's a depiction of the results from the Microns project, which was funded by both the Brain Initiative as well as by the U.S. intelligence agencies. And this was a characterization of about 1 cubic millimeter of mouse visual cortex, about the size of a grain of rice, again at synapse resolution, comprising 200,000 cells, over 500 million synapses, and 4 kilometers of axons. The static map by itself comprises 1 petabyte of data. Ultimately, we want to develop a whole mouse brain connectome at similar resolution, which is about 500 times larger. This will occupy about 1 exabyte of data. Even still, at this point, the researchers could obtain functional information on about 76,000 neurons and have used this information to build foundation models for how this part of the cortex processes information. So it's very exciting times for these projects right there. The third big project is the Armamentarium project, which has so far generated over 1,000 transgenic and viral vector tools for accessing brain and spinal cord cell types with exquisite precision. And as part of the ethos of the Brain Initiative, the the fruits of the labor from these large resource generating projects have generated open access data, over 12 petabytes and counting, as well as thousands of reagents. We already have about 3,000 entries into the AdGene database for dissemination for anyone who wishes to use it. And in terms of disseminating information, so far our groups have published 7 major publication packages in Cell, Nature, or Science, getting the word out to the research community, letting them know what's out there for availability for their use. And we have 2 more packages coming, I believe, in 2026. So these are very exciting times. Now, as you can imagine, we're generating a lot of data, and it's very important for us and for the field to manage that data. So we have been building a brain data ecosystem with domain-specific archives for these specific data modalities. We have 8 major archives. The brain data ecosystem isn't just big and growing, it's also smart. So we've built this network of specialized archives, each designed for specific types of neuroscience data. And right now, it's going to be all about organizing these data into data commons to build an actual knowledge base that can be used for further investigation and purpose-driven experiments. So I'll just conclude here by this has been a very, very brief overview given the time, but I just want to point out some opportunities for collaboration and cooperation between US and Japan scientists. Now, as mentioned before, the missions of the US Brain Initiative and the Japan Brain Minds 2.0 programs are highly complementary. And this is something I really look forward to seeing come to greater fruition, to see how these 2 programs at this level can find areas of synergy really to propel each other forward in their respective missions. But also at the more individual level, there's lots of complementary expertise and scientific interest among US and Japan-based neuroscientists and their home institutions. And I think that cross-fertilization of ideas will definitely be enabled by an open access data ecosystem. I think this is really going to be the key for global cooperation. And then finally, international collaborations are really needed to help solve the greatest challenges in neuroscience. Understanding how neural circuits and animal brains underpin behavior, I think, is one of the greatest challenges of our lifetime. Certainly, understanding this in the human brain is probably going to take generations to solve, and having strong collaborations and synergies across the Pacific, I think, will greatly accelerate these efforts. And you can learn more about the NIH Brain Initiative by going to brain.gov. And thank you so much for this opportunity, and I look forward to further discussion this evening and this morning.
Thanks so much, John. That's a fantastic talk on the brief, I know. Really exciting work. Okay. Well, thank you again, and please join me in welcoming our next presenter, Dr. Shigeo Okabe. Dr. Okabe is the project lead for the Japan Brain Minds 2.0 that you just heard a bit about, and director of the Riken Center for Brain Science. Dr. Okabe is also a professor at the University of Tokyo. Dr. Okabe, we're able to now see your slides. Thank you.
Okay, now you can see my slide. Yes. So, good morning. And my name is Shigeo Okabe, director of the Riken Center for Brain Science and project leader of Brain Minds 2.0. So I'd like to thank Dr. Benson and Dr. Isoda for providing me this opportunity. So today I will present an overview of Japan's national flagship brain science program and highlights its recent progress and also some future vision. So this program represents a major shift in Japan's approach to neuroscience toward a deeply integrated, model-based, and translational platform for understanding the human brain. So over the past decade, brain research has become a global collaborative effort. So we have seen large-scale national initiatives, such as the US Brain Initiative, the EU Human Brain Project, and also some major programs in Australia, Canada, China, and Korea. So these initiatives have transformed how we study the brain by accelerating technology development, data sharing, and international collaboration. So Japan has been an active participant in this global movement through Brain Minds program, and Brain Minds 2.0 builds directly on this foundation. So Japan's modern brain science strategy started with the Brain Minds. We started 2014, which focused on establishing a comprehensive understanding of the marmoset brain. This included multimodal circuit mapping, creation of genetic tools, and development of new imaging technologies. And later, from the 2018, Brain Minds Beyond expanded the effort to the human brain, particularly focusing on higher cognitive functions and psychiatric and neurological disorders. So together, these programs created both a prime foundation for the both basic and clinical neuroscience, which now converge to Brain Minds 2.0. So these earlier programs, Brain Minds and Brain Minds Beyond, produce several important resources. So first, a comprehensive marmoset brain atlas and data portal was generated, which integrates MRI, tracer-based connectivity, and gene expression, calcium imaging, and also some electrophysiology. So we also made genetically engineered disease models using marmoset, which can be used for the analysis of the Parkinson's disease and also the Alzheimer's disease. Also, we made several cutting-edge technologies, including wide-field calcium imaging and also the monitoring of the behaving marmosets. And also in Brain Minds Beyond, we have made the large-scale access of the human MRI databases by using the traveling subject methods. And also we created interspecies comparison method, which can be utilized to compare the human brain and the marmoset brain, and what kind of functional and also the structural similarities are preserved between these two species. So these resources now serve as the essential elements for the Brain Minds 2.0. So Brain Minds 2.0 started in March 2024 as a 6-year national initiative. Its central aim is to integrate multimodal large-scale datasets from molecules to behavior and to use these data to build mathematical models of brain circuit dynamics. The goal is not only to understand higher brain functions, but also to uncover the mechanisms of neurological and psychiatric disorders and accelerate the development of new therapies. The shift reflects Japan's commitment to predictive, model-driven neuroscience. So this is the organization of the Brain Mind 2.0. The Brain Mind 2.0 is coordinated by Weekend CBS, together with major partners, including the University of Tokyo, Kyoto University, QST, NCNP, NIPS, ATR, and OIST. And under the supervision of Professor Ryusuke Takahashi, this program consists of the core organization and 127 projects all across Japan. In addition, practical application support team supports the intellectual properties and ethics. So this structure promotes close integration between basic science, clinical medicine, computational modeling, and also industry. The Brain Minds 2.0 includes a remarkable broad network. Earlier Brain Minds program involved around 120 PIs, while Brain Minds Beyond included about 95. However, the current Brain Minds 2.0 program has expanded to 362 PIs and co-PI across Japan. So this includes researchers working on marmosets, clinical cohorts, computational science, and engineering. So the researchers are quite diverse. So such scale makes this one of Japan's largest coordinated national neuroscience initiative to date. So Brain Science 2.0 is structured around 5 key research themes. The first theme is innovative technologies and research infrastructure, including cutting-edge imaging technique, new probes, and multimodal data platforms. And the second theme is dynamics of higher brain function in humans. It is spanning neural activity, cognition, and behavior. And third part is pathological mechanisms. This connects animal models to the clinical research. And the fourth part is the digital brain. This is a quite unique approach. We want to develop computational models that integrate diverse data sets and that can be utilized for some prediction of the human diseases and also some animal behaviors. And part 5 is the series for the evolutionary diagnostics and therapeutics. So this enhances biomarker discovery linking to translational pipelines. So these things create a closed loop between technology development, biological discovery, and therapeutic innovations. So now I would like to use several slides to show some recent highlights of the Brain Minds 2.0. So on the basic neuroscience side, we first generated a Fragile X syndrome marmoset model, and this was published in Cell Reports this year. So this represents a major advance in primate disease modeling and opens the door to translational studies that cannot be performed in rodents. So this marmoset model actually shows several important symptoms, which are quite similar to the human patients. In infants, they show the altered vocal development, and in young adult stage, they show the alterations in social preference and also some abnormal auditory processing. And second, on the neurotechnology part, the development of the see-through cranial transparency technology was established. So this allows minimally invasive, high-resolution, long-term imaging without damage to the cranial bone. So now you can see the dendritic spine without making any damage to the cranial bone for a long time. So this was published in Nature Communications also this year, and this technique significantly expands the possibilities for live imaging in rodents and potentially in primates. And in clinical neuroscience, we have shown that unpuppet imaging has been revealed shared receptor level abnormalities across multiple psychiatric disorders. So here we can see that there are some downregulation of the AMPA receptors and also upregulation of the AMPA receptors in specific part of the brain. And this this pattern of the down and upregulation actually preserved in multiple mental disorders, including schizophrenia, bipolar disorder, and ASD. So this kind of technique highlights the value of molecular imaging-based biomarkers for the psychiatry and neurology. And on the computational side, we built a new neural circuit simulator using the entire Fugak supercomputer, which contains about 150,000 nodes to simulate the full mouse cortex at the biological detail. So this was conducted in collaboration with the Allen Institute and can be taken as a major milestone for Japan's computational neuroscience. So this presents the beginning of large-scale, biologically grounded digital brain modeling. We can start from the basic diagram of the neural circuits in the cortex and goes up to the local circuits properties, and finally end up with the whole cortex simulation. So looking ahead, BrainMinds 2.0 aims to build cross-species circuit frameworks linking mouse and humans. And also, we want to create predictive digital brain models that generate testable hypotheses. And third, we want to strengthen the pipeline from fundamental discovery to therapeutic development. And also, we want to promote open science, standardization, and global interoperability. So by integrating data, technology, and computation, we aim to contribute a comprehensive platform for next-generation brain science. So in summary, BrainMinds 2.0 represents a major step for Japan toward integrated, scalable, and translational neuroscience, tightly connected to global initiatives. And thank you very much for your attention. I look forward to future international collaboration.
Thank you so much, Dr. Okabe.
Thank you.
Now my pleasure. And please remember to place your questions in the Q&A chat, and we will share them with our speakers following the presentations. And you'll have an opportunity to hear their response. It is now my pleasure to introduce Dr. Aaron Gittler, the Stanford Medicine Basic Science Professor in the Department of Genetics at Stanford University. And Dr. Gittler is also a recent BRCP awardee. Dr. Gittler.
Thank you very much, Naomi, and thank you to my colleagues for the opportunity to share our recent work and the support of the program for a workshop that we've designed, which I'll tell you about based on the science I'm about to describe. So in my laboratory, we're studying the neurodegenerative diseases, frontotemporal dementia and amyotrophic lateral sclerosis, or FTD, and ALS. And the hallmark pathology in these diseases is the mislocalization of an RNA binding protein called TDP43 from the nucleus to the cytoplasm, where it forms these aggregates out in the cytoplasm, and it's depleted from the nucleus. And a major effort in the field, including in my laboratory, is to try to figure out the molecular consequences of the loss of this nuclear RNA binding protein. And pioneering studies have identified that when TDP43 is normally in the nucleus, it binds within introns and represses sequences that are so-called cryptic exons. And following splicing, these cryptic exons are left out of the mature messenger RNA. However, when this TDP43 function is compromised by its loss from the nucleus, these cryptic exons creep in to the mature messenger RNA transcripts, and they disrupt the reading frame, these destabilize the RNA, and in some cases they can even make cryptic peptides, which have shown promise as biomarkers. So the hunt has been on for identifying the cryptic exons that are formed in these neurodegenerative diseases. Major breakthrough came in 2019 when two teams led by Don Cleveland and Clotilde Giortaren and another group led by Kevin Egan discovered that STAthmin2 is a gene that harbors a cryptic exon that's regulated by TDP43. And when you lose TDP43 function, this causes a truncated STAthmin2. In my laboratory, we set out to try to identify additional ones. And we did so by interrogating a published data set of neuronal nuclei sorted from the brain of patients. And we discovered, using our reanalysis of RNA-seq data and looking for alternative splicing, we discovered 65 alternative splicing events that occur in neurons from individuals with TDP43 depleted from the nucleus. And we verified STAthmin2, but then we found a whole bunch of new ones. And the exciting one that we focused on is called UNC13A, which I highlighted here in red. This was exciting because UNC13A is known to the ALS and FTD field because it's the #1 genome-wide association hit if you don't include the chromosome 9 locus, which we now know encodes C9orf72. UNC13A keeps coming out as a major genetic contributor, but no one knew how that could relate to disease. We found in functional studies that if we depleted TDP43 from neurons and then measured the UNC13A cryptic exon, abbreviated CE here, we see when only in the cases when we lose TDP43 function does the cryptic exon get inserted. This results in a loss of the normal transcript and then a loss of the protein UNC13A. UNC13A is a critical protein involved in synaptic transmission, and it's deleterious to lose its function from neurons. We then went on to analyze human patient postmortem samples and found in every neuron in which TDP43 was lost from the nucleus, we see abundant UNC13A cryptic exons. So we think that this is a new facet to TDP43 pathology. And then excitingly, I mentioned this was a genome-wide association study hit, and it turns out that the lead GWAS SNP is right in the cryptic exon itself. This immediately suggested a mechanism by which these SNPs might increase risk for disease, perhaps by making the cryptic exon more prone to be included when TDP43 function is compromised. And indeed, we tested that hypothesis and found in patient cells that were heterozygous for the risk and the protective allele, there's more RNA-seq reads harboring the UNC13A risk allele. And then in a dose-dependent manner, we see as you go from 0 to 1 to 2 risk alleles, you see more and more cryptic exon inclusion in the brain. And perhaps most excitingly, we see that if you just look at survival of individuals stratified based on their risk allele, there's a decreased survival if you have 1 risk allele, and even further if you have 2. So we're excited because we think if we can correct this single cryptic exon event, it should have a powerful rescuing effect. So we published these studies alongside a parallel team led by Michael Ward at NIH and Pietro Fratta at University of College London. And we think we've defined a new facet to TDP43 pathology, and we're very excited to look at these additional cryptic targets we've defined. We're looking at if genetic contributors to cryptic splicing may promote or protect against these. UNC13A is the first one. We think there'll be new ones. Therapeutic strategies we're trying to pursue to prevent this from happening in the first place. And in recent unpublished studies, we've extended beyond UNC13A, and we've been functionally studying additional cryptic splicing targets, and we have a preprint studying these. So we think it's turning out that more and more of these are involved in neuronal function and synapse biology, and we're trying to figure out how that function and dysfunction connects to neurodegeneration. In a new unpublished work, we realized very recently that cryptic exons are not all created equally. Stathmin-2 makes a mature messenger RNA that's stable, but UNC13A cryptic exon destabilizes the messenger RNA because of a process called nonsense-mediated decay. And this is a molecular biology process in which the cell has a quality control to make sure you're not producing aberrant RNAs. And when the ribosome is translating an RNA, it kicks off this exon junction complex that's deposited at exon-exon junctions. If there's a premature stop codon, this ribosome stalls and this exon junction complex does not get kicked off, recruiting a complex that then degrades the RNA. That gave us this idea that if many, perhaps most of these cryptic splicing events we were identifying induce nonsense-mediated decay, maybe the standard RNA sequencing approaches we had been using would have been missing these. In other words, could there be even more cryptic exons out there? So we did a very simple experiment where we just inhibited nonsense-mediated decay and repeated our RNA sequencing analysis. And the big surprise came where we found hundreds, maybe even 1,000 novel cryptic exon events that were masked and that by nonsense-mediated decay. And the one I'm most excited about, this is again unpublished, is called DNMT3A. It encodes a de novo DNA methyltransferase. We never detect it normally. When we inhibit nonsense-mediated decay, we don't detect it. But when we inhibit TDP43 and nonsense-mediated decay, we now unmask this cryptic exon. Importantly, when we go back and we look at the protein levels, we can see that DNMT3A was always down all along, and just using regular RNA sequencing, we just missed it. So we're very excited about this expanding portfolio of cryptic targets for FTD and ALS. And we didn't just stop at cryptic exons. There's 3 actually preprint study describing these results from us and other teams. And in recently published work from 5 independent groups, we found that beyond cryptic splicing, TDP43 regulates another facet of RNA metabolism called alternative polyadenylation. In other words, where the poly A tail is added. So given this convergence of RNA processing defects, alternative polyadenylation changes, cryptic splicing, their impact on neuronal biology and cryptic splicing, we think it's the perfect time to convene a workshop of our colleagues from the United States and Japan to study convergence of RNA processing alterations and synaptic and neuronal dysfunction in neurodegeneration. So with the generous support from this program, we're delighted to announce a workshop coming in summer of 2026 in Osaka. My colleague, Professor Yuka Koika from Niigata University, and I are hosting this, and we've been invited a number of leading researchers in synapse biology and RNA processing biology from Japan and United States. And I'm very grateful for the support and excited to foster further interactions and collaborations. Thank you.
Thank you very much, Dr. Gettler. Certainly look forward to the workshop. Please note that your camera seemed to have turned off. I don't know if you were aware of that, or if it was just because you were sharing your slides, but if possible, please do turn your camera on when you're able. And thank you again for your presentation. Okay, and now very happy to introduce our next speaker, Dr. Tomomi Shimogori. She is the Deputy Director of the Riken Center for Brain Science and Vice Project Lead of the Brain Minds 2.0. And Dr. Shimogori is a former BRCP awardee by the NIDS. Dr. Shimogori, are you able to hear us?
Yes. Can you hear me?
We can, clearly.
Okay, great. Thank you very much. I have to apologize that for some reason I cannot turn on my camera, so I can't show my face. But can you please share my slide?
Yeah, we'll do so now. Lois, could you pull up her slides, please? Thank you.
Great, thank you very much. So in case I had my picture on the right side of the slide, so now you can see how I look like. To thank for this opportunity to introduce our a little bit old work, but I would like to show how it actually connected to my recent work. So thanks to this program, I was funded from 2010 to 2013. As I said, it's a little bit while ago. And I worked together with Seth Blackshaw at Johns Hopkins. And how we did the collaboration is basically we were both interested in the diencephalic development. So he was actually pulling out all the genes using genomic analysis and a lot of different time points of the mouse hypothalamus. And I did all the validation using in situ hybridization to find specific gene which are expressed in a specific hypothalamic nuclei. So next slide, please. Okay, so I guess I don't have to talk about like what's the function of hypothalamus for this audience, but as you can see in this slide, the hypothalamic function is quite diverse. However, they are playing really important fundamental role for the brain function, such as like sexual diversity and sleep, wake cycle, and circadian rhythm, controlling the body temperature, so on. And because they have a lot of interesting functional features, we wanted to work on it, but then we found like, there's quite a lot of limitation. One of the main major limitation is we couldn't manipulate specific nuclei at the specific timing. And because of the lack of the molecular markers which label the specific nuclei at the specific timing. So this is the reason why we decided to work together to figure out if there's any specific marker gene which we can label specific nuclei or ablate or manipulate from the very early time point. Can you go to the next slide? Right. So again, I told you this is a little bit old work. So this is before like the novel technology like a single cell RNA sequencing or spatial transcriptomics. We had to do like a microarray analysis, and for the validation, we did in situ hybridization for every single molecule. So CES started to collect the samples from very early embryonic stage, such as E10.5, all the way up to like postnatal day 40, and collected the sample and then isolated the possible marker gene. For the first validation, first round validation, we started from over 1,000 probes and then tested a lot of different developmental time point and first checked like whether these probes are working. And so we isolated 1,000 positive probes. And we went for the second round of validation using in situ hybridization. Here we selected 352 probes, which show like a specific regional expression pattern in the hypothalamus. And from here, we use like a coronal and sagittal section from 11, 12.5, 14.5, and 16.5. And also what we realized that because the hypothalamus changed their location during the development, for instance, they changed the orientation almost 90 degrees. So it was very difficult to follow which cells are moving to where. So here we decided to add another marker gene like a sonic hedgehog as a landmark to make sure that we are looking or we are following the same populations. Next slide, please. Yeah, so here's like a quick summary of how it looks like. So here's like the diagram of the E12.5 mouse hypothalamic structure, and on the bottom right side of bottom is a real in situ data with the 2-color in situ. So here we're using the variety of the marker gene, but now you can actually tell how dynamic it looks like. So on the right side of the screen, like in the D, you can find that there's a little hole in the blue region. So this is just not the error, because if you do another in situ hybridization using different marker gene like LHX9 and figure G, you can find that this marker gene perfectly fits into this whole region. So here, using lots of combination of the marker gene, we found that how the hypothalamus are making their structure during the early developmental time point and which cells are moving to where. And then we also could figure out from which cells are coming from which part of the hypothalamic ventricular zone. So with using these kind of really strong, the area marker gene, we could also analyze the mutant animal. Next slide, please. Right, for instance, this is like a mutant of the sonic hedgehog, and since the sonic hedgehog was we were using as a landmark, but it like I already showed you, it is expressing in the ventral part of the hypothalamus. And when you knock out hypothalamus, then on the right side of the screen, this is how it's going to look like. So in the wild type, it is showing like we could identify lots of different region of the hypothalamus, but in the knockout, when you just look at the structure by simple staining, it just looks like completely messed up. But using the specific marker gene, you can actually find that most of the ventral part of the hypothalamus is really screwed up, but like the dorsal part is quite remain in there. So you can, with using these kind of marker gene, you can actually find these kind of abnormality even from the very early stage of the development. And next slide, please. So like using this kind of, marker gene, what you can also do is you can look at these marker gene expression in different species and do the comparative analysis. So starting from the lamprey here, it has a little bit more simpler structure. However, you can find using the marker gene which part of the hypothalamus is conserved all the way to the mouse. So like here, you can actually do lots of different things with these kind of, like I'm usually calling these kind of little animals. But next slide, please. But we also want to look at these kind of bigger animals, such as primate. And even in the primate, as you can see on the left side of the screen, their structure are quite different, and then size is also very different. And on the very left side of the screen, it's really tiny, but this is the size of the mouse brain. So if you want to look at the human, or human and non-human primate brains, mouse is actually not good enough because their size is so different, and there's a big jump in between these like different species. So if you need to have lots of different model animal to do like a complete comparative analysis to find which part of the brain is conserved or not conserved. And one of the largest gene expression atlas is like in Allen Brain Atlas, which has mouse, macaque, and human gene expression. However, even in the non-human primate, you can see like there's a huge differences in between these species. Maybe it's better to have another species or like a non-human primate species in between to do better comparative analysis. So we decided to use common marmoset in the red circle here for another model animal. Next slide, please. So this is how we started our generating our marmoset gene atlas. So it was actually part of the Brain Minds project, which was introduced by Dr. Okabe here. We started like 2014. And then it actually run for 10 years. And here what we did is we actually used a developing common marmoset brain and look at like over 2,000 gene expression. We also generated the function that we can find the gene expression in a specific brain region. So after it is finished at 2023, we luckily could have the second project, which is Brain Minds 2.0, and we are still continuing looking at the gene expression in the common marmoset. But now we are also using like a different technology like single-cell RNA sequencing or the spatial transcriptomics, which will be released on this website sometime soon. Next slide, please. Yes, within this project, what we also want to do is, because now we are dealing with lots of different, gene expression. However, like if you look at the different marmoset brain, their size and their the shape is slightly different. So if you want to compare gene expression in different animals, then we cannot overlay to each other and then see whether these are the same cell types or not. So what we have to do to make us able to do that kind of comparative analysis is we have to generate the standard brain and put the old data inside of that, so that we can actually compare different brains. So here, this is what exactly we are doing right now. So we have a standard brain space, and for each brain sections, we collect like a Nissl staining or the backlink, and then make sure that they all fits in the standard brain region, and we put back the gene expression into this. So on the right side of the screen, and now you can see that we can actually compare the different types of gene expression on the same standard brain region. And also using this, we can actually put the single-cell RNA sequencing data, which actually don't have much spatial information in it. But however, we already have this marmoset spatial data. We can actually compare those single-cell RNA sequencing data into the spatial information, so we can precisely tell which genes are expressed in which part of the brain. Moreover, we can actually collect the single-cell RNA sequencing data from the human brain, and using the marmoset brain, it actually helps us to predict how these human gene expression look like inside of the human brain. So if we can generate like the human brain structure inside of the digital space, we can put all of these data together and predict how the human brain looks like. And also, since we have lots of developmental data, it actually helps us to predict how the gene expression looks like in the future or in the disease status. Since we have lots of knockout marmoset for the mental disorder or neurodegenerative disorder, we also help us to predict how these disease model gonna develop their disease status inside of the brain in the future, and also we can actually predict how it's going to look like in the human brain. So this is our goal for the Brain Minds 2.0, which I'm hoping like we can actually get this sometime soon. And then if we can open this, people can throw your data in and actually help you to also, look how your data looks like in the human brain. Okay, next slide, please. So this is pretty much it. And I'd like to thank all of my lab member and also the funding. Thank you very much.
Thanks so much, Dr. Shimagari, and hopefully next time we'll be able to see you virtually as well. Terrific presentation. So thank you. We will now, and thank you to all of our presenters. And we will now transition to the open discussion portion of the webinar and invite everyone to please place your questions for our speakers and presenters in the chat. And we will review them, answer them as soon as they appear.
Dr. Meredith has her hand up for a question.
Please go ahead, Dr. Meredith.
Yeah, hi. I wanted to comment on the last talk. Dr. Shigemori, your data were so beautiful and really spatially precise, and it made me wonder if you were able to consider time of day in this project, because the hypothalamus contains the circadian clock, and most of it is so strongly circadianly regulated. Just want, not that I want to add another 10 years onto your workflow, but I'm just wondering if there was any consideration for that, because I imagine these cascades change by time of day.
Yeah, that's a really good point. And I guess that's also really important for the, especially for the postnatal animal. So when we were looking more like at embryonic stages intensively, because, you know, we care about the developmental stage, we were always collecting the, you know, the middle of the day. So that's the only time point I can actually say. But otherwise, I mean, I don't know much about the you know, the clock genes and the embryonic brains at this moment. But I think that's a really important point, especially when we do like a, you know, marmoset, we are doing a lots of, you know, postnatal animals analysis. So I think that's a really important point. We should put like what time of the day we collected the day, you know, the sample on our metadata, maybe. Yeah, thanks for the question.
I'd love to talk to you about that further. That's actually my area of research.
Okay.
Thank you.
Thanks.
Thank you. Yeah, that's wonderful, Dr. Meredith. Yeah, this positive outcome of this meeting today. Please raise your virtual hands or put your question in the chat, and then we will, and you kind of mute yourself. Dr. Little.
Yeah, I just wanted to say when I click on the chat, it says that it's turned off for this meeting.
Oh dear. Let's
Sorry,
see.
you have to go to the Q&A.
Okay.
I did.
Q&A box.
Yeah. But please go ahead, Dr. Little, one of our NIH colleagues supporting the program. Feel free to voice your question.
It's more of an observation. And I mean, hearing what these two large programs are doing, Brain and Brain Minds 2.0, it seems like we should have some further communication about where there could be synergy and where there might be opportunities to do things with this vast amount of data that we have that maybe we couldn't do alone. So I just wanted to suggest that.
That's a really great point. That's why we're really excited that we were able to get all of our excellent speakers. Again, congratulate you, particularly Dr. Nye and Dr. Akabi for your presentations. And we'd like to get you to work together. And that really highlights the purpose of our program.
Yes, I agree. And in fact, Shimoguri and Okabe senseis did host me for a very, very nice visit this past July at Riken. And we had great opportunity to interact. And also I got to meet a number of really exciting scientists at the Japan Neuroscience meeting in Niigata. So these we will try to push this at both the institutional level, but also at the personal level, because this is where these relationships get formed and pushed forward. So thank you for that comment.
Yes, I'm also willing to collaborate with the US Brain Initiative. So we also already talked with Dr. Nye about the possible collaborations, and we had very nice conversation with him. So we want to push this kind of direction forward.
Dr. Meredith and then Dr. Eugenen.
Yeah, I just wanted to make a quick comment on the heels of Dr. Little's comment. One of the areas that I see is ripe for synergy is that we're all inventing the same data ecosystems to house these really large data sets and to federate them. And the international accessibility and interoperability of that is really critically important. So I would say at NINDS, that's certainly something that we're very much interested in partnering and working on joint initiatives. We have some that are in collaboration with Brain, but we also have some that are in collaboration with other large-scale programs. And I think the development of secondary use studies really drives the design of the ecosystem. And I'm really interested in learning more about those secondary use studies that you have going on in your programs. And we can talk offline about it. But I definitely want to identify that as a specific area of shared interest where we could maybe pool or consolidate resources and really get to a better place. Thank you.
Feel free to contact us, either Stacy or Naomi, and we can help make some of these connections if you don't have some of the numbers. Dr. Eugenen and then Dr. Little.
Oh, thank you so much for the presentations. Really, really nice. And thank you so much to the NIH and the Japan agencies for putting this together. It's amazing what that happened in these 10 years. Then is any interest, because I see that NIDA is also connected to a study, not only the normal brain and some genetic diseases, related diseases, but also drug abuse. And in addition to that, we have the visit of some Japan agencies here in Texas with the interest to study some encephalitis virus, especially attacking the brain. Most of them emerging in Asia and that area. I would like to have some comments about it. Thank you.
Yes, thank you for your comments. And we Brain Minds 3.0 also interested in both drug abuse and also some virus infections. And recently we published a paper about the connection between the COVID-19 infection to some change in the AMPA receptor distribution in the brain. So that kind of direction should be quite important for us.
Dr. Little, if no other comments about that question.
Yeah, I also want to thank the speakers. They were fantastic talks and very compelling. The using the marmosets as a model for development is really fascinating, but it also occurs to me that it's also a resource coming from a nerdy Evo-Devo perspective to look at the differences between what is known about human development and marmoset development and other primate development to begin to understand the species-specific aspects of early brain development. And so I think this is an exciting aspect of perhaps this collaboration, and that it could be something that could be explored for, this is an area of synergy between our two projects, I think. So it could be an interesting place for just taking my comments earlier one step further.
Yes, thank you for your comments. We really appreciate, and we are putting much effort for the marmoset brain, but we also have to provide more data for the human brain and also the other non-human primates brains like the macaque. So that's a kind of huge effort and cannot be done only by the Japanese initiative. So really hope to have some kind of synergy with the US Brain Initiative. Thank you very much for your comment.
Thank you, Dr. Little. And we have a question from Dr. Takahashi for Dr. Gittler. First, great talk. What is your estimate of the number of cryptic exons that are suppressed by TDP43?
Great question. So our initial study, we found about a little bit less than 100. And then in this new study with unmasking them using inhibiting nonsense-mediated decay, that is in the hundreds to even 1,000 range, depending on how you count them. So I think there will probably be several hundreds of these. And moreover, there are going to be some that are cell type specific, like specific to neurons. And then there'll be some that are glia specific and some that are general. And we're trying to figure out what makes one, and then some will be stronger and weaker. So it's a very exciting time, and that that's going to be one of the topics of our workshop, is to try to get a priority list of these and then functionally study them. Thank you for the question.
Any other questions? Please let me know if I'm missing any in the Q&A, or raise your virtual hand to be recognized. It's late in the US and early in Japan. So no other questions, then yeah, just thank you for Dr. Gidler and Dr. Takahashi. No other questions. We want to respect everyone's time because of the lateness in the US and early in Japan. I think we're, and Stacy and Naomi, correct me if I'm off. I think right now I'm to thank everybody for attending. Okay, all right. Thank you for attending the meeting today. The goal of this
Richard,
meeting
we're
was
just to hand it over to Dr. Issa when you're ready to provide some closing remarks.
Yeah. Okay, can you see me? And can you hear me?
Oh, okay.
Yeah, okay. Yeah, yeah.
You don't mind, Richard?
Yes. No, please go ahead. Yeah,
and then we'll turn it back to you.
Okay.
Thank you, Dr. Issa. It is an honor and a pleasure to introduce Dr. Tadashi Issa, which many of you know, the Director General of the National Institute for Physiological Sciences in Japan. Please, after you, Dr. Issa.
Okay, so yes, thank you for your introduction. And I'm Tadashi Issa, the Director General of the National Institute for Physiological Sciences. I'd like to make a closing remark. Thank you everyone for attending this webinar session. Unfortunately, we could not make the annual usual BRCP meeting at the Society for Neuroscience meeting. So instead, we planned an alternative meeting by including the introduction of the flagship projects on both sides. That is Brain Initiative on the US side by Dr. Nai, and Brain Minds project on Japan side by Dr. Okabe, in addition to the presentation by awardees of the BRCP grants, Drs. Jitra and Shimogori. I truly believe that today's webinar was very fruitful and successful for exchanging the information about the current status of neuroscience on both sides. And we could know how active interaction is ongoing by the leading scientists on both sides based on the BRCP funding. So this webinar meeting yielded a great outcome than the usual meeting. So I hope this outcome will lead to the further strengthening of the mutual interaction and collaboration between both sides. So in the end, I would like to express my sincere thanks to the great contribution by the NIH staffs, Dr. Benson, Stacy Chambers, and Naomi Buka for making this meeting possible. So I hope to meet you all in person in the near future again. Well, thank you very much for your attention. Thank you again.
Thanks so much, Dr. Issa. Very well said. And over to you, Dr. Benson, to close this.
Thank you, Stacy. Yes, I agree. So thank you, Dr. Issa, and thank you to all of the speakers, Dr. Okabe, Dr. Nai, Dr. Shimogori, and missing Dr. Gidler. Yes, thank you as well. So please feel free, if you don't have the contact information for anyone, please contact our office, and we're happy to make those connections. We happy to continue another 25 years of this collaboration with the US and Japan. So thank you, and have a good morning and a good evening, everyone.